everyone. My name is Regan Volk, and I'm a third year in the Zara lab at UCSF in the CCB program. Today, I'm going to be telling you about my work, proteomic characterization of human monocyte-derived macrophages. Macrophages are like guardians of the body, removing aged or mutated cells and infectious bacteria or viruses. They perform phagocytosis, where they engulf or eat and digest target cells. There are many therapeutics currently being developed which aim to encourage macrophage phagocytosis, leading to remediation of cancer or infection. One of the ways macrophage phagocytosis is regulated is through small chemical signals. In the case of my work, this small signal is called interferon gamma. When resting macrophages are exposed to interferon gamma, they become stimulated and active. You can think of it like drinking a coffee to wake you up in the morning. The stimulated macrophages are better prepared to phagocytose and do their job. However, not much is known about what changes occur in the macrophage upon stimulation, especially at the protein level. Proteins are molecules in the body which are responsible for doing every sort of task you can imagine. In my research, I wanted to answer the question of what happens at the protein level that allows stimulated macrophages to phagocytose better. I took patient blood samples, isolated their monocytes, and derived them into macrophages. Next, I split the macrophages in two and treated half of them with interferon gamma, our small chemical signal. I then used mass spectrometry, a powerful tool that allows us to identify proteins within a cell to determine all the proteins present in the stem versus unstem macrophages. I did this for 3 separate donors and found that globally, the macrophages had similar amounts of protein, but the stem had unique groups of proteins they were enriched for. As you can see on this bubble plot here, there are a couple pathways which the stemmed macrophages showed a bias toward. On the plot, the size of the bubble indicates how many proteins were associated with this pathway. The fold enrichment tells us how enriched they are in the sample, and this is shown on the x-axis. And finally, the color indicates how significant this is, with lighter color being greater significance. We saw that stem macrophages had more proteins associated with engulfment of cells during phagocytosis, cell recognition so they can identify their targets better, and immune response so they can notify other cells there may be an infection. My research has been able to show what important changes are occurring at the protein level in macrophages when exposed to chemical signals like interferon gamma. The hope is we can use this knowledge to our advantage, encouraging macrophages to eat more, especially in the case of cancer where this can remediate tumors. My future work will look more closely at how macrophages respond to exposure to cancer cells, and if there are proteins present that could be viable therapeutic targets. Thank you so much for your time, and I hope you enjoy the symposium.